First, I would like to thank the Scientific Committee for the kind invitation and the opportunity to share in the first electronic conference on medicinal chemistry some experience with aminations of small molecules to obtain bioactive compounds. My name is Sousa. I'm a professor of medicinal chemistry at the Faculty of Pharmacy, University of Porto, Portugal. So the idea is to give a perspective of generating bioactive amines from a medicinal chemist's point of view, highlighting the strengths and the weakness of each approach. And this is the abstract of my talk that is available at the website. Why are amines so important to us medicinal chemists? This group offers both hydrogen bonding properties and the potential for increasing compound solubility, together with simple and wildable applicable methods of forming a CN bond. Not surprisingly, alkylation and arylation of groups with nitrogen are in the top 10 reactions used for preparation of drug candidates. And why xanthones? This benzogamma-pyrrone scaffold can be found in a large number of natural products. With diverse biological activities, is considered a privileged structure, attractive from a synthetic and structure-activity relationships point of view. The aromatic rings provide the opportunity of displaying binding functionalities from a rigid core in numerous vectors. In our group, we follow a common approach of most medicinal chemists, of synthesizing a common core motif and then perform multiple derivatizations of this core in order to generate useful SARs for a given project. So we have two ongoing projects of E2 lead optimization where we pursued the amination of xanthones, with pendant functionalities not always the ones that favors reactivity. And the aim of this talk is to share the most reliable transformations that furnish the desirable compounds, but also the ones that did not. Briefly, in the first project, we intended to develop dual anti-tumor and P-glycoprotein inhibitors based on leucanthone, a clinical candidate for cancer therapy, and we do that by merging the thioxanthone scaffold with the requirements that we know are important for PGP inhibition. In the second project, we followed an hybridization strategy based on known inhibitors to develop new disruptors of this protein-protein interaction, p53 MD-M2. The first project involved the annealation of an available chloropropoxy thioxanthone. We followed a structure-based design and built an homology model of PGP. To select the most promising molecules, we used docking studies of these ligands that were previously filtered by some known rules like Lipinski into the transmembrane and the nucleotide binding domains of this protein. To obtain these target aminated thioxanthones, we apply a classical Kupper-mediated cross-coupling reaction. With the first conditions investigated, using copper oxide, the reaction was only successful for simple aliphatic amines. For poor nucleophilic and high-hindred amines, most remain unchanged, albeit the reaction time was prolonged. What we saw was a major product that was this methoxythioxanthone from competing hydroxy attack on the aryl chloride. The addition of potassium carbonate in these mild temperature conditions provided the necessary deprotonation step, and we were able to obtain more 2 amines. but only modifying the heat source to microwave at harsh temperature conditions, and the solvent to N-methyl pyrrolidone, an aprotic alkaline solvent more appropriate for microwave heating, a moderate conversion was observed and the reaction proceeded cleanly. In some cases, a mixture of NMP and water was needed to improve the solubility of the alkaline catalyst and the amine, allowing the reaction to proceed under these conditions. So the amination reaction worked not only for alkyl amines, but also for cyclic and aromatic amines. Since we needed a heat compound in amounts for in vivo studies, we further investigated this reaction in milder temperature conditions to develop a reliable and scalable process. We examined the effect of several copper species and loading that are frequently used for copper coupling reactions, and a relation of xanthone using copper iodide produced higher yields compared to reaction using other copper sources. To avoid competition of CO cross-coupling, several bulkier alcohols and other polar aprotic solvents were investigated, but the yields were dramatically lower. We also examined palladium catalysis, which was not successful either. The development of ligands for the coupling of less reactive substrates, such as aryl chloride, prompted us to screen several ligands, and this highlighted here, NEN-dimethylglycine furnished the most efficient catalytic system. We followed a not so rational approach when we tried heterogeneous catalysis with a clay. I think we were desperate at the time. But the fact is that interesting results were observed and they deserve to be further explored in an organic chemistry perspective. So after scaling up, in optimized conditions, the free base was converted to the pure hydrochloride salt through a crystallization that purged this byproduct. So, the utility of any relation is no doubt aided by the easy preparation of the halogenated precursor and also of enabling techniques such as microwave and catalysis. In our particular case, obtaining directly the hydrochloride avoided any chromatographic step. Nevertheless, it seems plagued by poor substrate scope and the striking requirements to ensure that only vanishing small levels of residual alkylating agents and metals should be present in drug candidates. Concerning the target molecules, also there is a potential for in vivo toxicity, since they are anaromatic with risk of metabolizing to anylates. So the second project consisted in developing a library of Aminated Xanthones by the one-pot conversion of our wheat compound that we call Lem2 using a reductive amination. We had already experience in this discovery of inhibitors of P53-MTM2 based on this scaffold with interesting results both from natural and synthetic products. We originally obtained the heat compound, aldehyde, by a Duff reaction, but it was from a pathway not suitable for scale-up. So we decided to develop a more desirable strategy and plan obtaining the aldehyde from the appropriate monobromomethyl precursor. What happened was that instead of one bromo group, we obtained mainly the dibromo derivative. So we went to search in the literature and we use the liquid ionic 1-butyl-3-methylimidazolium tetrafluoroboronate for the selective debromination to what we thought the monobromo derivative. Surprisingly, we directly obtained the desirable aldehyde in a very clean, high-yielding and fast reaction. And we believe that this follows a solvolytic displacement. Finally, we proceeded with the reductive amination protocol with solid supported cyanoborohydride in methanol, and what we obtained was the alcohol. Herein, we found that the choice of the reductive agent and solvent are critical to the success of the reaction. Just to highlight, Lem2 only reacted in THF and STAB to reduce selectively the imines over the aldehydes. And with this protocol, the yields were quite satisfactory. So, I should highlight in this protocol the advantage that from a medicinal chemistry perspective, I emphasize that these aliphatic amines have low potential to form reactive metabolites. Also, these were metal-free reactions. As a keynote, I hope I could show you our chemical toolbox that allows us to select the best synthetic protocol to furnish the desirable chemical transformations. I would like to emphasize the importance of enabling techniques in synthesis, such as microwaves, new solvents like ionic liquids, and catalysis. Moreover, with this approach, we reduce the use of solvents due to alternative solid phase extractions and allows the development of high-throughput purification methods. Finally, I should like to thank, first, unexpected results that were seen as enabling techniques also, and gave us some improvements in these experiments. Of course, the entities that were responsible for the financial support of this work, and I hope you enjoy this conference and you enjoy this day.